So this is something that should be familiar to us all. We have talked about the sets of 3 nucleotides that code for an amino acid. So this is the same thing as we were seeing with those nucleotide or codon charts. It's just in a different format. It's organized by amino acid rather than organized by the nucleotide sequence or pattern. And there are a couple things that I want us to take away from this slide. So one is the title here. The genetic code is redundant, which means that there are multiple codons or multiple triplets sets of 3 nucleotides which code for the same amino acid. There are very few amino acids that only have one codon that code for them, and one is methionine, and that is a special case, and the other one is tryptophan, and that one is not such a special case, but it does just have one triplet that codes for it. The other thing that I want to point out is that there is something else called a stop codon, and this does not code for any amino acid at all. And in addition, so we're going to be reading the nucleotide triplets to see what the ribosome is going to do with the mRNA. So it's going to read a triplet and it's going to attach the appropriate amino acid. And the other thing that I want to point out here, even though it's not labeled, unfortunately, is that the AUG is also a start codon. And that means that it's basically the complement or the opposite of a stop codon. So it begins translation, and it also calls for methionine. So that's important to remember because students often get a little bit confused about this because the stop codons don't call for any amino acid, but the start codon starts and it calls for methionine. So just keep that in mind. All right, so again, we have this code, and then we have these amino acids. And we've been saying, oh, these triplets call for an amino acid, but we haven't said anything about how that actually happens. And we'll talk about that now. So we need to get from a nucleotide triplet to an amino acid in order to make a peptide sequence, a peptide chain. How do we do that? Well, this is done through the use of tRNA or transfer RNA. And transfer RNA in your textbook is referred to as an adapter. And I think that's really appropriate. I think that's an excellent description of tRNA because it interfaces with 2 different systems of language or systems of information, basically. So the comparison I make is if you've ever traveled abroad or if you're from another country and you came here to the US, you might have had trouble using electronics because here in the US we use this system with the 2-pronged flat plug, and physically it just doesn't interface with this system, which is the European system where you have the 2 round prongs. And I know that in addition, underlying this, there are also electrical differences, but we're just going to talk about the physical differences right now, because that's all I know about. So obviously you can't just plug in your cell phone charger or your hairdryer or whatever to a European outlet. What you need is an adapter. And so you could buy these really unwieldy, expensive things at airports, but they're really important. They're crucial, and these are adapters. And they have interfaces to work with both of these systems. So they have a place where you could plug in your American version of your electrical plug, and then they can also interface with the European format for electricity. And this is the same thing that we have the tRNA doing, because again, we have a similar problem wherein we have nucleotides. We have nucleotide codons, which can't interface with our amino acids directly. but we need some sort of adapter to make sure they both come together appropriately, and this is tRNA. So, this is a cartoon of a tRNA. We have the amino acid at the top, and we have something called an anticodon. Now, the anticodon is going to be the part that interfaces with the codon on the mRNA, and the amino acid is the amino acid. And so this is how we get the bringing together, the interface of these two information systems. This is a slightly more realistic version of a tRNA. It looks very similar, but it shows the actual chemical structure of an amino acid, which is bound to the top of the tRNA, and then it also shows our anticodon down here, and the anticodon is going to form hydrogen bonds through complementary base pairing with the mRNA. All right, so we're calling this tRNA. What is tRNA? Well, it's made up of RNA, and it's made up of RNA that is folded, which we haven't talked about before. We've talked about linear RNA, which is what we use for mRNA. So this tRNA is folded. This is the 3D structure of a tRNA. It looks a little bit complicated, but if you flatten it out and look at the 2D structure, this is what it looks like. So you can see the code for the tRNA here. There are some strange nucleotides that you might not be familiar with, like this one here, the Y. There are a few of them. Those are just uracil, U nucleotides that have been modified. And so this RNA is transcribed the same way that mRNA is transcribed, but then afterwards it's folded, and once it's folded, some of the base pairs are modified. Those uracils are modified so that it stays folded. All right, so if we have folded tRNA, what happens next? So we just have this bottom part, we don't yet have the amino acid. How do we put the amino acid on there? Well, this is a really crucial step that many people end up forgetting. And this is the action of a tRNA synthase. A tRNA synthase, also shown over here, is an enzyme that can bind to a folded tRNA and an amino acid. So it is sequence specific for the anticodon. And it is shape and chemistry specific for that amino acid. And this is how you get the appropriate amino acid attached to the correct tRNAs. Again, this is called tRNA synthase, and it is an enzyme or a protein, so it's made up of amino acids. And the uncharged tRNA, or the tRNA that is folded but does not have an amino acid attached, will be bound here, and then this tRNA synthase will form the bond between the amino acid and the tRNA. And the bond that is formed is an ester bond. All right, so now hopefully we have a better appreciation for the structure and function of tRNA. And next we're going to talk about another type of RNA, and that is the ribosome. So the ribosome is a really important structure in every cell. Every single cell, no matter what domain, has ribosomes. And they're all fairly similar. We could recognize similar structure, similar sequence, and of course, the same function all across all of the domains of life. And ribosomes are made mostly of ribosomal RNA. So again, RNA that's transcribed the same way that mRNA is, but it is folded so that it has a 3D structure. And in addition, there are some proteins that associate with this rRNA to help them stay folded. There are two parts of a ribosome, so the large subunit and the small subunit, and that is true also for all domains of life. Now I want to specify that although there are proteins which are part of the ribosome, the proteins don't have enzymatic function. They're purely functional. Structural. The proteins are just there to help maintain the structure of the ribosomes. Experiments have been done that will remove the proteins or make them non-functional, and you just have the rRNA, and the ribosome still catalyzes the translation reactions. That's not true if you remove the rRNA and just have the proteins. And so this is really interesting and important, and you guys will do a reading about RNA and the origin of life on Earth. And we think that RNA is very ancient and probably more ancient than DNA because RNA can have a catalytic function and DNA does not, at least so far as we know. All right, so as a functional unit, the two subunits of the ribosome come together, the large subunit and the small subunit, and there are binding sites for the mRNA as well as tRNA molecules. First, I'll show you the 3D structure here. So this is the 3D structure of a ribosome. It's a little bit difficult to see. The lighter top part is the large subunit, and then the darker green part down here is a small subunit. And then these three colors in here, which you can hopefully see, are the places where 3 different tRNAs can bind, or the 3 different places where 1 tRNA can bind, depending on how you look at it. And this is a depiction of that in cartoon form. So we see the 3 sites, which are called E, P, and A, or A, P, and E, right? Whichever way you want to memorize it is fine. The order actually in terms of function is APE, so maybe you want to memorize it as APE instead of EPA. And then the mRNA binding site is just a channel through the small subunit. So we'll talk about how this works, how the whole system recognizes the correct tRNA as it reads the mRNA and adds an amino acid. A couple things we want to note beforehand. The ribosome moves down the mRNA 5' to 3'. So that is the opposite direction of the reading of the polymerases that we talked about so far. So keep that in mind. And in addition, without the ribosome, the cell can't do anything. It can't make any proteins. So if you inhibit the ribosome, which some poisons or in some medicines do, then the organism really can't continue to function. It can use the proteins that it already has, but it can't make any new proteins, which is really crucial for a cell to carry out its metabolic functions and maintain life. And that's why the ribosome is being kind of salty here. All right, so before this whole process of translation is carried out, the ribosome actually needs to be assembled. So this involves the large and the small subunit as well as mRNA and also one single tRNA. In addition, there are a couple of proteins that are important. These are called translation initiation factors. This occurs only in eukaryotes. So first, the small subunit binds to these translation initiation factors, as well as a methionine tRNA. The methionine tRNA is called an initiator tRNA, and it always binds in the middle site. And it always binds in that middle site, which is the P site. I'm going to take that here. The P site and the A site and the E site are empty at this point, and we don't have the large ribosomal subunit. All right, so next we add the mRNA. So the mRNA can bind to the small ribosomal subunit when it's bound to the translation initiation factors, which also associate with the mRNA, and then the methionine is going to interact with the mRNA as well. However, the anticodon for the anticodon for this tRNA, the methionine tRNA, is also going to match up with the start site, which is the AUG. And at that point, that's when we have kind of like the magical combination, which is going to trigger the recruitment of the large ribosomal subunit. So let us go back for 1 minute and take a look at the composition of a mature piece of mRNA. So we have that 5' cap. That can associate with the translation initiation factors. Then we have this non-coding region. And we also have a coding sequence, and then another non-coding sequence and the poly A tail. So what we're talking about in translation initiation is the 5' cap, and then we have a non-coding region, which is where the small ribosomal subunit and the tRNA are going to initially bind, and then they're going to move down to find the start codon here. So let's go back to that image that we were just looking at. So here we have, again, the anticodon for methionine matching up with the AUG, and the methionine anticodon is going to be a UAC. And this triggers the release of the translation initiation factor and the recruitment or the binding of the large ribosomal subunit. And so at the end here, we're going to have the large and the small subunits bound to the mRNA. The AUG is in that P site, and the methionine tRNA is also at the P site. So this is really important because it's unique. There's no time during the elongation phase of translation where you're going to see a tRNA in the P site but not in the A site. And there's no time during elongation where you're going to see a tRNA incoming and initially bind to the P site. This is totally unique to the initiation. All right. So why is it initial for translation to begin in the correct place on the mRNA? So take a moment and think about that. And I apologize if you could hear a lot of background noise. I have the AC going, and my husband is working in the other room. So why is it vital for translation to begin in the correct place? Well, if it doesn't, then you can get a frame shift mutation in the resulting protein. Because if you don't start directly at this, if you shift one nucleotide down, first of all, it's not going to match up with the methionine anticodon, but also you would cause a frame shift for the reading of the rest of this mRNA. And if you shifted over 3, then you'd be losing an amino acid. So you're going to change the structure and probably the function of the protein if you don't begin at the start site. So what is up with these letters that name the sites? So the technical names for these, the A site is the amino acyl site. The P site is the peptidyl site, and the E site is the exit site. I use these words to associate with them because I think this is a bit easier. The A is the arrival site, so the arriving tRNAs go to this site. The P is the poly site because that's what is going to often hold the polypeptide, and the E is the exit site because that's where the tRNAs are going to leave. All right, so translation elongation. Again, this is how it occurs in eukaryotes, but it's very similar in prokaryotes. So charged tRNAs, or that is tRNA molecules that have an amino acid bound to them through an ester bond, come into the A site, the arrival site, or the aminoacyl site. We've already passed the initiation phase, and in this image they show some amino acids that are already forming a peptide chain. You could also imagine this as the methionine tRNA, and this is right after we ended initiation. Either way, the same things are going to happen. So we have a tRNA in the P site, we have a new tRNA in the A site. So the next thing that occurs is the formation of a bond between the amino acids in the P site and the A site, and the destruction of the ester bond between the amino acid in the P site and its tRNA in the P site. So this is the enzymatic function of the ribosome, which again is due to the RNA, not the proteins. And again, that is the formation of a peptide bond and the destruction of the ester bond. After that, we have the shifting of the large ribosomal subunit. And so that occurs kind of as a result of the end of this making and breaking of bonds. You get the shifting of the large ribosomal subunit. And so if we're paying attention to only the sites in the large ribosomal subunit, now we see that the tRNA that just was in the A site, or it was the new one a moment ago, so this blue one, the one with the 4 on it, that was the incoming one, which was, it still has the intact ester bond. That is now in the P site. The A site is empty, and the original tRNA is in the E site. So this was the one that was there previously when this blue 4 tRNA arrived. So the E site is the exit site, and it's really not large enough or chemically structured to hold a tRNA for long. So that tRNA is going to be unstable and is actually going to pop out of that site. So this orange 3 tRNA, which is now uncharged, is going to leave that site. And as that happens, this small ribosomal subunit is going to shift, and it is going to shift so that it matches up with the large ribosomal subunit. So let's look at that one more time, the comparison here. The large ribosomal subunit, I want to point out as well, is now hovering with the A site over this dark blue codon. And now the small ribosomal subunit is going to shift so that the A site matches up with that dark blue codon set as well. So now we see this guy has been ejected, kicked out. Where does he need to go? This 3 orange tRNA that is uncharged needs to find a tRNA synthase that can accommodate it for the appropriate amino acid and the appropriate anticodon. And now we have this blue tRNA, number 4, that is in the P site. What's going to happen next? Well, the whole thing. We're going to get a new tRNA coming into the A site. The anticodon is going to match up with this codon on the mRNA, and then the ribosome is going to break this bond between the number 4 and its amino acid and form a bond between number 4 and number 5, and that is going to be a peptide bond. And then the large ribosomal subunit will shift, and this number 4 will get kicked out. Then the small ribosomal subunit will shift to match up with the large ribosomal subunit. And so it's kind of like the circle of life. It just happens repeatedly for dozens or hundreds of new codons, anticodons, and making up all those peptides. So you're getting a peptide chain which is forming, and again, these look like just circles, but they are different amino acids with those unique side chains, which are going to help the protein fold correctly after it's all translated. Or I guess it can also start to fold during translation. Now, how do we end translation? That is when the ribosome comes across a stop codon. Now, a stop codon doesn't code for a tRNA that's associated with an amino acid, as we see here. Instead, a stop codon is going to call for a different type of molecule. So there are 3 different stop codons. They are used preferentially by different organisms, and there's some variation in actually what triplets code for stop codons. So don't be surprised if you come across that type of idea in the future. But for our class, we're just going to talk about these 3 very common stop codons. And the stop codons, as I said, they actually match up to something called a release factor instead of a tRNA. And that is shown here. So here we see a stop codon, the UAG, and that is going to be bound by something called a release factor. So that's this red thing. And the release factor is only going to come into the A site, and then it's actually going to change the activity of the ribosomal, large ribosomal subunit to break the ester bond, but not bind or not create a peptide bond. And so this is actually done through the adding of water, it says in your book, which just means that it is, you can't break this bond without stabilizing that atom with another bond. And so that's where the addition of water comes from. And this, again, this only happens due to the initiation factor being here in the A site. So after that process occurs, you're going to have, just pretend there's not even any dots here, you're not going to have the ester bond here between the tRNA and its amino acid. So there's really nothing holding this peptide chain in the ribosome anymore. So then we're going to get the dissociation of the peptide from the ribosome. We're also going to have the dissociation of the large and the small subunit, as well as the dissociation of the small subunit from the mRNA and the release of the release factor, as well as the tRNA. So everything in this is going to kind of fall apart. They're all going to go their separate ways. The peptide chain is going to go and be folded appropriately. The tRNA is going to go off and be recharged by tRNA synthase. The release factor is going to float around until it's needed by another ribosome, and the mRNA can be translated again, or it might be degraded. And then, of course, the 2 ribosomal subunits can come back together at the appropriate time through initiation with another mRNA, and they'll go through translation again. So ribosomes are used over and over again. They really don't have any downtime. They work really hard. They're definitely essential workers, and they are just reused constantly. All right, and so that's our process of translation. Now I want to talk just really briefly about the differences between translation initiation in eukaryotes and prokaryotes. So we already talked about eukaryotes, but now we need to talk about prokaryotes. And one of the differences between these 2 systems is due to the modifications of the mRNA that occurs in eukaryotes. So recall again, in eukaryotes, they have this 5' cap, which associates with these initiation factors, which help the mRNA bind to the small ribosomal subunit. In prokaryotes, they don't get that 5' cap because they don't need to leave the nucleus. However, they still do have a 5' end, and the 5' end actually retains the triphosphates here. Now, why does that happen? That is because only the incoming nucleotide is going to have the 2 phosphate groups removed. So if you go back to the transcription slides, you'll see that the initial nucleotide is just kept as it is, the next incoming ribonucleotide is going to have 2 of the phosphates removed. So then you get 1 phosphate, and then the sugar group of the 3rd ribonucleotide can come in and bind to that. But the first nucleotide hasn't had those phosphate groups removed. So I think that it's really helpful if you are kind of struggling with that to just draw it out, and it'll become more clear. So for even the prokaryotic mRNA, we have those 3 phosphates, which are going to indicate that that is the 5' end of an mRNA. And then in addition, we have these sequences called ribosome binding sites. The ribosome binding sites are upstream of the start site. So we see here the AUG, that is a start site. And then upstream of that, we have a binding site. We also have this non-coding region between the 5' end and the ribosome binding site, and then we also have a non-coding region here, 3' end. Now, this particular mRNA reflects something that we talked about yesterday, which is the fact that prokaryotes can have these polycistronic mRNAs, which means that they contain multiple genes in a single piece of mRNA. And that is because these genes all have some functional relationship. They're all part of the same metabolic process or something. And so they need to be transcribed together. And they're regulated together, so they all share a promoter on the DNA, and they all share a transcription stop sequence, or transcription, a transcription, what word am I looking for? We'll just say the end of transcription code, or termination sequence on the DNA. But then when we make the mRNA, they have individual RNA binding sites and AUG, the start sites. And this is because they can each have a ribosome binding to them and translating at the same time. So you could actually have one ribosome starting here at the same time as one ribosome binding here and a 3rd one binding here. And so we get translation of these proteins really quickly, and you don't have to wait for the first protein to be translated for the second protein to be translated, which is really neat. So again, instead of looking for that 5' cap with the initiation factor, the small ribosomal subunit and the tRNA in prokaryotes will look for those ribosome binding sites. And so then you get the same type of assembly after the binding there occurs, and the elongation process, as I mentioned, is extremely similar, or pretty much identical to that of eukaryotes. All right, so that is translation. I do want to mention one other thing, which I think is really relevant in our world, if not for the particular epidemic that we're going through right now, then maybe slightly less acute pandemic that we've been going through for many years, and that is that of antibiotic resistance. And antibiotic resistance is a real problem in the medical world, as many of you probably know, and there's a lot of components to this. Some of them are due to the misuse of antibiotics, some of them are due to the difficulty in discovering new antibiotics. Regardless, I want to mention how certain antibiotics work. So antibiotics target only bacteria because they target bacteria or unique molecules that only bacteria have and eukaryotes do not have. And so that doesn't mean that they target molecules that don't exist in eukaryotes, but the molecules need to be unique or different enough in prokaryotes so that they don't bind to the eukaryotic version. And so this is true of prokaryotic, or particularly bacterial ribosomes. So I mentioned previously that all organisms have ribosomes, but that they're recognizable across domains. So interestingly, if you ever go into research to study new bacteria or to try and identify bacteria from the environment, you may likely look at something called the 16S sequence. That is part of the ribosomal sequence in bacteria. And the 16S sequence is part of, or is a sequence that we can use to compare all different species of bacteria, or at least many different species of bacteria. And we could also compare that sequence to sequences in eukaryotes, because the ribosomes are similar enough, even in sequence, that we could see the evolution and the relationships in the sequence throughout the tree of life, which is pretty neat. However, okay, so I talked about all the similarities, but there are definitely differences, and the ribosome in prokaryotes is a different size. The two subunits are different size and slightly different shaped than the ribosome, the ribosomal subunits in eukaryotes. And the difference in size and the difference in shape allows us to make antibiotics, and also for bacteria to make antibiotics that target only the bacterial ribosome and not the eukaryotic ribosome. And so some antibiotics will target the ribosome and stop translation throughout the entire cell. And as I said, if a cell can't perform translation, then it's not going to be functional. It can't make any proteins. It's really, it's like past the point of a struggle bus. It's like, well, you can't replace anything. We really need some reinforcements here, right? Bacteria don't usually get reinforcements when they call for them. But here we see something called the 50S subunit, and so that is the large ribosomal subunits in bacteria. The 30S subunit, which is the small ribosomal subunit in bacteria. Now, I mentioned the 16S sequence, which is 16S subunit, which is a part of one of the other subunits. It's just a small section, but I don't want to expand too much more on that. What we are looking at here is the assembled ribosome, and then there is this molecule, which is an antibiotic called azithromycin. There are also similar antibiotics, erythromycin and clarithromycin. You can see that they are very similar structurally, and they will all bind to the large ribosomal subunit, the 50S ribosomal subunit in bacteria, and they stop the transferase activity, which means that we could get a tRNA coming into the A site, but you're not going to get the transfer. You're not going to get the formation of a peptide bond between the A site and the P site, and the tRNA cannot move from the A site. And so effectively that stops translation. It blocks translation. And this molecule is not going to dissociate from the ribosome. It binds permanently to the ribosome, effectively stopping all translation after it initially binds. As an aside, I do want to acknowledge also that we only see the A and the P site here. The E site is here, and so it's kind of inferred because it's the exit site. There's nothing in it for very long, and so it's not really shown as part of the large ribosomal subunit. So again, this is an example of one type of antibiotic which targets the function of the ribosome in bacteria. And again, it only targets bacterial ribosomes because of the size, shape, and chemical differences between the bacterial and the eukaryotic ribosomes. Great. So now we've talked about transcription, which is transforming this information in DNA into mRNA, and translation, which is transforming this information from RNA into protein. We also, in the past, talked about DNA replication, which is DNA to DNA, and in the future, we'll talk about PCR, which goes from DNA to DNA, or could even go from RNA to DNA. So our last slide here is a bunch of different terms that you should be familiar with. So some of these we went over in today's lecture, and some of them we've gone over previously. If you don't know any of these, I encourage you to look them up in the book. One I'm seeing here that we did not specifically talk about is the wobble base, and so I'll go over that right now as our last little tidbit. So a wobble base is the third base in a triplet, and you'll notice, for example, if we just look at alanine here, the codons that code for alanine all start with different sequences, or the same sequence, sorry, but we could have the third nucleotide can be anything. So if you have a G and a C, doesn't matter what the third nucleotide is in the codon, we're going to get alanine. So similar here for asparagine. So we have a G and an A, whether the third codon or the third nucleotide is a C or U, you're still going to get the same thing. And we see this for many of the amino acids. Glycine, similar, as long as you have two G's, and any other nucleotide is going to result in a glycine. So this is called the wobble base, and it's part of the redundancy of the genetic code. It is important because, I have this, I don't know if you guys could see this, but it's important because it's advantageous in an evolutionary sense. If you get a mutation in that third base pair, it doesn't matter if it mutates, as long as you have the two first base pairs in the codon. The other one, let's throw away. You can have it mutate to anything and it'll be fine. And so this helps to prevent any effect of a mutation on the protein. And in addition, we've talked previously about another way that the protein might not be affected by a mutation, which is that you might have a similar amino acid being coded for due to a mutation. And if the amino acid is similar in size and chemistry in its R group, then that might also not affect the function and structure of a protein. All right, so I think that is everything. And again, take a look at this last slide. I will upload these slides to the website, and have a great day, everyone.